Thanks a lot for the kind introduction. Thanks very much to many people here in the audience who made it possible that I come to Stazione Zooologica Anton Dohrn, especially Christiane, Angela, Andrea, Wiebe. I hope I didn't forget anybody. It's a special opportunity for me, I must say. I'm not used to speak in marine biological stations, but I have spoken in one this year, earlier this year in Helgoland, and I come back to that. It's a German island. You understand me? Okay. I come from Dresden, which is between Berlin and Prague, halfway, in the former east of Germany. And we have actually our cathedral built by a Roman architect. And we have some influences of Italian architecture in Dresden, also patterns. However, I will not speak about urban architecture, more about biological architecture. And thanks also for the idea to bring back some pieces of my work. Is there any people in the audience who have a mathematical background? Nobody. Okay. Then don't worry. I will keep the technical level of my presentation low and want to share with you problems in which I'm involved. Should you be interested in any of the problems in more detail, we can definitely talk after my presentation. I will be here until Friday this week. Yes, and I chose to focus on organization principles of biological systems. And one way to start is to ask what is life. I'm not the first to ask the question. I'm also not the last to answer the question. I want to share with you some important aspects of life. One is reproduction, which is present in life, obviously, and it ensures species continuity. Another aspect you might think about, and this was mentioned already in the introduction, pattern formation. This is an extraordinary property of living systems, and as we will see, it's not in any sort trivial. How can it be that living organisms spontaneously produce? Don't worry. Anton Dohrn had no PowerPoint. So in case it doesn't work, I will continue verbally. Yes, I brought some patterns which come from the sea. And another aspect in front of the building, collective migration, in this case of organisms. And the question is, how do these forms emerge? And finally, evolution. This is really something which is not shown by non-living organisms. And it allows that populations diversify in large time scales. And there's many more. So these are properties, and these are definitely not all properties. You might work on other properties of life. But these ones I want to focus in my talk. And the question is, how are these processes organized? The key point of my presentation is that organization principles, new insights in biological questions are preceded by development of technology and theory. First, I would like to focus on reproduction and share with you the insights Nathanael and Pringsheim received in the context of sexual reproduction in algae. He could do this because in the beginning of 19th century, microscopy evolved And there was a cell theory by Matthias Schleiden and Theodor Schwann. Pattern formation is due to collective migration and evolution. This study because there was the possibility for mathematical models and computers. And we will see in my talk how this influences each other. So we'll start with Nathanael Pringsheim, who you can find here in the archive. There are letters by Pringsheim to Anton, Dohrn. And we have them yesterday already digged into them. There is two main types of reproduction, asexual and sexual. And you might think that sexuality is an old term. Where did the term originate, you know? In botany. And we owe this term Linnaeus. In Systema Naturae, he suggested we are going for which problem? The connection with the outside world. Okay. Okay, continue. Okay. Okay, Linnaeus suggested a classification based on a sexual key. And this means he classified plants according to the position of sexual and male and female sexual organs, stamens and pistils. And this is nicely shown here. We see 23 classes which look very similar. These are the so-called seed plants. And there is a special plant class based on spore reproduction. He called these here publicae, nuptiae coram totum mundum visibilem aparte celebrantur. Weddings are celebrated visibly in the public, in the presence of the whole world. And the last one, at that time, one didn't even know if there are weddings. Nuptiae clam instituunta, weddings are celebrated secretly. This choice was actually provoking protest of many botanists. They didn't like this associations with weddings. For example, this class here is called polygamy. Johann Wolfgang von Goethe and many others protested. However, the sexual reproduction of these plants here, the seed plants, was accepted in the beginning of the 19th century. It was open what happened in this little special class, which contains the ferns, mosses, and algae, so-called lower plants. Beginning of 19th century, it was clear that ferns and mosses have a sexual reproduction. What was unclear if algae have it. And it was, we owe to Nathanael Pringsheim that he showed first the sexual organs in action. And this was in a yellow-green algae, Vaucheria sessilis. It's a microalgae, very small. And what we see is the female organ containing the egg. And we have the male organ, which produces spermatocytes, which reach the egg. And this was very much provoking, and he got a lot of critical feedback. Other botanists didn't believe, and
maybe he has not cited other works sufficiently. So he continued his research. And then 1 year later, he saw first time fertilization in a green algae. And this is an Oedogonium species. This is particularly neat because the males are so-called dwarf males. They sit on the side of the female organ. And this is an original drawing by, sequenced by Nathanael Pringsheim. And we have actually made this sequence of drawings alive. This is the egg. The male dwarf, spermatocytes, reaches the egg and fertilization. So this was then also agreed by the critical botanists that this is a fertilization. And green algae is particularly neat because green algae are the ancestors of all land plants. To see the evidence of this finding, this is a temporal scale, 1 million years ago, formation of solar system, prokaryotes, blue algae, eukaryotes, multicellular green algae, and we on top only very small, tiny. And this fertilization found by Pringsheim is actually a universal principle, so that sperm cells migrate to the egg cell is a universal principle, and we owe this to our sea ancestors. So most of the evolution took place in the sea. But we owe to Pringsheim another finding. He also showed that there is an asexual sexual alternation of generations. This was first found in Tunicata by an Italian, Camisso, and it had been established in mosses and ferns by the middle of the 19th century. And Pringsheim showed that algae also have an alternation of generations. This is the asexual sporophyte, and on top we have the sexual gametophyte. By the way, you needn't write down anything. I can share also the PDF of my talk. And this has a long-lasting implication. If we can namely look at the sexual evolution of land plants by the variation of the alternation of generations. This is from green algae all the way to gymnosperms and angiosperms. And what we see in the beginning, the algae, they have a gametophytic dominance. The sporophyte is very small. And then finally, the higher seed plants, there is a gametophytic, a sporophytic dominance, and the gametophyte is very small. So this finding in the green algae has a lot of consequences. And Pringsheim not only showed this in green algae, but he also contributed a theory, which is still discussed today. Yeah, well, who was this Pringsheim? He comes from a Jewish family from Silesia, which migrated to Silesia in the 18th century. And there is a lot of branching. This is him in the age of 35. He studied first philosophy and medicine and shifted to botany under the umbrella of medicine. His father was a wealthy iron plant owner, and he wanted his son to follow this, but he didn't like this. And under the umbrella, he could study his favorite botany. He bought a very expensive microscope, took drawing lessons, and from then on, he basically, in the rest of his life, he was drawing and doing microscopy. He was very much influenced by Schleiden and Schwann's cell theory. This is from his PhD, 1848. And this is a quite costly, chic microscope, which costs roughly, at that time, the annual salary of a professor. He was the founder of German Botanical Society. Last week, we had the annual meeting. This society still exists. It's today international. And he was also the founder, initiator of the first German marine biological station on the island of Helgoland. Yeah, as we know, we are in the Stazione Zooologica, which was first independent European Marine Research Institute, opened by Anton Dohrn. Before he had done this, he was traveling in Europe and looked for a good position for such a station. And he also was in Helgoland, Germany. And this picture, you might know, it's actually also downstairs in the aquarium. And we have two Italian biologists. This is the German one from University of Bonn, and we have Anton Dohrn and Ernst Haeckel at that time together. Yes, and it took a long time until Germany got its own station. In 1890, Helgoland becomes German. Before it was Danish, and in the last century, British, and Germany exchanged Helgoland with Zanzibar. Pringsheim quickly realized the chances that Helgoland became German, launches an initiative, and the institute was already open 2 years later. And this is the first building, and you see there is obviously some difference. There was a change in the architecture, but the institute still exists today, but it has never reached the niceness and aesthetics of the Napoli station. Who knows where Helgoland is? It's in the North Sea, Hamburg here, roughly 1,500 kilometers from Napoli. Here's the Netherlands. And Helgoland is not only important for biology, marine biology, but also for quantum physics. There's a book by Carlo Rovelli, you know it, Helgoland, Italian physicist. And this is due to that on Helgoland, Werner Heisenberg found the foundations of the modern quantum physics. So it's not a travel guide to Helgoland, but it's on the history of quantum physics. So Pringsheim can definitely be called a pioneer of lower plant botany. He not only did research on developmental cycles of algae, but also on mosses. This is, for example, Hypnum and ferns. And he was the first scientist in the Pringsheim family. The grandchildren generations then had to suffer from Hitler. They either were deported, committed suicide, or had to emigrate. And one grandson of Nathanael Pringsheim, Rudolf Ladenburg, was also a founder of quantum physics. Okay, that was, but this is by the way, that was Nathanael Pringsheim in a nutshell. And now I would like to shift gears and go to pattern formation. Patterns are abundant. Spatiotemporal patterns are anywhere, but it's the idea how these patterns can be formed is only some years old. And we owe this idea Alan Turing. Alan Turing, he was a mathematician, one of the most important mathematicians in the last century. He did very key work in computational problems. Unfortunately, he died already 54. He was homosexual and committed suicide. And he not only contributed to computer science mathematics, but to biology. And this is a key paper, 52, Chemical Basis of Morphogenesis. And in this paper, he combined an idea, namely to diffusion, transport diffusion, which normally destroys patterns, diffusive processes. When you start with a concentration gradient, diffusion destroys. But he combined it with a particular reaction kinetic. So the first sentence says everything. It is suggested that a system of chemical substances called morphogens reacting together and diffusing through a tissue is adequate to account for the main phenomena of morphogenesis. What does the Turing idea say? We have an activator and an inhibitor, morphogens. Activator activates its own production, it's the production of inhibitor, and inhibitor has a negative influence on the production of activator. We have decay. That's the simple reaction kinetics. And this he combines with diffusion. So let's look at the temporal development from top to bottom. We have a one-dimensional line of cells. Each faces a certain concentration of the activator inhibitor, steady state, and then we put a little bit more activator due to its own activation. We get growth, we get production of inhibitor. And now we have to assume that inhibitor diffuses quicker, faster than an activator. And then we get new activator maxima in some distance. And if you think this idea further, you get a spatial periodic pattern. And this is the idea of Turing. It looks simple, but it is having a lot of implications. And this was a cute idea to couple a non-pattern formation mechanism with a special kinetics. And if you think that a certain threshold is needed for cell differentiation, then you get periodic patterns of cells, for example. Yeah, the model, sorry, this is a mathematical formula. It's a so-called partial differential equation. We have concentrations changing in time. This is the diffusion part and the reactive part. You needn't worry about the details. Hans Meinhardt from the Max Planck Institute Biology in Tübingen, he applied these chemical equations to biology. And this shows a simulation of organizer formation. So you can see there are many parameters in the system, and by changing parameters, initial conditions, and boundary conditions, you can get many different types of patterns. It took 50 years to get a proof of a Turing pattern in a biological system. First, they have been identified in chemical situations. And there are many variations. I just wanted to show you the key idea of a pattern formation mechanism. And now I go to collective migration. This is the prominent example. And how would you define such dynamics? There are two scales. We have the individual microscopic scale, and we have the population, the macroscopic scale. And the key idea that this population behavior emerges from interactions of the individuals. That's the idea of collective migration. So, and the individual has not inbuilt any picture of the swarm. It's a bit similar to a traffic jam in front of the building. Studying the engine of the Fiat does not help us to understand traffic jams. They are brought about by the interaction of the cars, acceleration and stopping. So it's the same idea. This collective migration does occur on many scales. This is from very small macromolecules, over organelles, cells, tissues, and organisms. And my work is focused on the cell level. This is an example of endothelial cells which produce network patterns. This is the focus of my own work. And the question is, what do mathematical models help in order to understand this? Yeah. So this agent can be a cell, an organism, a molecule, and we want to know which interactions lead to what types of patterns, migration patterns. So we are interested to identify the rules of interaction. So, and this means we need a cell-based mathematical model. And this allows me to introduce the prototype of a cell-based mathematical model, namely cellular automaton. We owe this to John von Neumann, who was interested in the theory of so-called self-reproducing automata. John Conway, he invented a computer game in the 70s, and due to the availability of computers at that time, one could run heavy simulations. So this is an ecological model. We have living cells or dead cells. A living cell can survive, can die, or can give birth to another cell, depending just on the number of living neighbors in this 9-cell neighborhood. That's all. And this simple game is actually computational universal. And what happens in the next time step? This cell has not enough living neighbors, so it will die. And in this case, the same will happen. But if we start with this configuration, this and this will die, but there will be birth, because this and this cell have exactly 3 living neighbors. So what we get is an oscillation. And this is an example of an emergent pattern. The oscillation is not visible at the level of the rules. Prototypic example of emergence. So it's a deterministic cellular automata. Depending on the initial condition, the fate is given. And this is just an example. Mathematicians like simplifications. So this is 2-dimensional. What is the simplification of 2D? 1D. So we put everything in a line. Now each cell can only have left and right neighbor. And there are 8 possible neighbor configurations. 2 to the power of 3. And for each configuration, we define if the new state is 1 or 0, dead, alive or dead. And this is an example of a temporal development. And this rule here nicely resembles shell patterns. And this rule is the rule 90. And this is the decoding of the rule. And now we can start from 1 cell. And we get our shell pattern. We can look at other rules. Let's look at the traffic rule. This rule can model 1-lane traffic to the right. It's all online available, so you can check yourself. So this is the most abstract cellular automaton. What I want to show you, although it's so abstract, you can find applications in nature. In this model, there's no migration. But particularly in cellular systems, we have migration. So we want a model where we have the cellular automaton idea, discrete agents, and migration. And we take the idea from fluid dynamics. Navier-Stokes equation is describing particular regimes in fluids. And this is one of the most difficult to solve equations in applied mathematics. It was a big surprise that in the 80s, these authors found a very simple automaton rule which can simulate Navier-Stokes equation. And we stole the idea, we couple the classic cellular automaton with a fluid dynamical cellular automaton. So we have this we have a channel structure in each lattice node. we can change configuration, and then each agent moves in the corresponding direction on the lattice. That's all. The dynamics here is, again, the state space. So at each lattice node, we have this channel structure. And in one time step, we can have burst-test processes shown here in Sketch. We can have switches, genotypic, phenotypic switches. We have reorientation, and then the propagation. This is all. There's a Wikipedia page available for this bio-LGCA and also an online simulator. One can mathematically formulate this, but this is of less interest. Again, you needn't look at the formula. Just a sketch. This is a configuration, and we want alignment. Alignment means that this agent tends to the majority of the neighbor directions. So with a high probability, because the neighbor direction is mainly to the right, he should turn to the right. And this can be formulated in terms of so-called transition probabilities. Don't be shocked. Important, we only have in the model 2 parameters, namely the density of agent and the sensitivity to the neighbors. If the sensitivity is 0, we have random motion. I show you now a simulation. These are channels, channel occupations. Let's go to a hexagonal lattice. And let's assume that the sensitivity is 0, and we expect diffusion. No? This is a temporal development, and in fact, no pattern formation. Now we make the sensitivity a bit larger, and we get swarms of alignment. It's a nice aspect. Oop. The nice aspect of this type of models is you can not only run simulations, but also a mathematical analysis. I will skip the technical details. This is the density, and this is sensitivity. Unstable means swarms, stable no swarms, and you have to cross a certain critical density or sensitivity to get swarms. And this you can see in the simulations. This is a mean orientation, which is a kind of proxy for the swarming tendency, and you need a certain sensitivity to get swarms. Again, you might think these models are too simple, but they have applications, in this case, to myxobacteria, where you can get these swarms, and we can predict the critical sensitivity and critical density. This is a real Myxococcus, and this is from a simulation. Yeah, that was my example of collective migration. Again, I've chose the most simple idea for collective migration, and one can extend the simple idea, and we have done this to incorporate more factors, but this was the ingredient. And last but not least, I would like now to turn to evolution. Here in the Institute, you're probably most interested in evolution of species. I am interested in a different evolution, namely of cancer. So tumors also are characterized by evolution. So everybody has malignant cells, no? And they mutate. And if they manage to survive, then they build clones. And the big question is, how does this process work? And this is the focus of my work. How does this heterogeneity of clones emerge? And I would like to show you again a most simple idea. So we have proliferation only in our model and random walk. And the mutation is applied to the proliferation rate. Whenever a cell gives birth to two new cells, then the daughter cells have a slightly changed proliferation rate. That's our mutation. We have death and we have the random walk. And this is again our hexagonal, our lattice gas. The different colors indicate different mutations. And the question is, how does the dynamics look like? This is a one-dimensional situation. We start with a mutated cell on the left side. This is the cell density, and here is the mean proliferation rate. And what we see in time, that the largest proliferation occurs at the tumor edge. And this is known as gene surfing. This is an observation which comes from the simulation, and we try to understand this. And again, don't be shocked, there will come a mathematical formula. We can approximate the dynamics for the density by a partial differential equation, which has cell death, logistic growth, mutations. And the mutations here are mathematically a diffusion, mixing. And this can be further simplified. We neglect space and only look at the temporal development of the density. And this is actually a fundamental theorem of natural selection, which is known for 100 years. But with our model, we underpin this macroscopic law by some ancient dynamics. Yeah, so this can be compared, the simulations and the approximations in time, both for density and proliferation rate, and they have a nice agreement that shows that our mathematical analysis can grasp the main ingredients of the computer simulations. And we can do this and put this in 2D. This is the local cell number. Blue is high, yellow, blue is small and yellow is large. And here the largest proliferation rate is along the edge. So what we have already seen in the 1D simulations. And you might again think this is so simple, but there is a possible application in tumors. This is a mouse, and the distance of the tumor start and the tumor hotspot is actually an indicator of prognostic value. So it's interesting to find out where the hotspots of proliferation are. Yeah, I would like to turn to the end, summarizing. I tried to introduce you shortly to the work of botanist Nathanael Pringsheim. And if you are interested in further details, there is a book which will come out in November 2024. And I've actually prepared a list. Should you be interested, then I can send you an email if it has appeared. I wanted to bring it here, but the production process is delayed. I'm sorry. There is, however, a German version, so you can get an idea how it's structured and about the figures. There's also a calendar available for 2025, which shows the original drawings by Nathanael Pringsheim, who was not only a botanist, but also an artist. Yes, and the 3 other examples, you should check a book on cellular automata, which appeared already in the 2nd edition. Again, I try to stress that all these insights are preceded by technological findings, microscopy, computers, mathematical modeling in particular, and I try to give you some examples. We do also work on cancer invasion, immunobiology, cell jamming, and even on coral morphogenesis. The simulator from which I chose the simulations is available online, and this is an example of the activator inhibitor system, and I want to finally show this nice pattern forming with you. So we have two species, activator and inhibitor. You see the coloring over here and the formation of patterns. And I can tune the distance between the maxima and the pattern by changing the difference of the diffusivity of activator and inhibitor. And if I'm not cautious, then I can show the simulations only every 100th time step. But watching fish, you need cautions, so I just wanted to. Yes, and finally, thanks a lot for your patience. And I'm looking forward to questions. Grazie.
Well, thank you very much. That was 4 talks in one. And yeah, the connectors are clear. So questions?
Thank you for your talk. It was very interesting. And so I have 2 questions. The first is very simple. How did you get to know about Nathanael Pringsheim work? And if there are some archives in the city you're living in, how did you step into Nathanael Pringsheim? And the second is, you were speaking about a model where you actually model sensitivity and density of individuals in a population is something I can figure out, but the sensitivity I couldn't understand, which is the biological significance of this parameter.
Thanks. The first question was how I got involved into Nathanael Pringsheim. Yeah. Okay, so I'm interested in history of ideas, scientific ideas, since my studies. My first PhD offer was in history of mathematics, which I denied at that time, since I thought maybe I go into biology first. And then later, my aunt has the funeral speech of Albert Einstein for the uncle of my uncle. And he is Rudolf Ladenburg, a grandson of Pringsheim. I didn't show the pedigree here because I wanted to focus. It's not an aunt by blood, an uncle by blood, yeah, important. But however, he's the uncle. And then I got interested because this Rudolf Ladenburg, I found in information that he worked together with John von Neumann. And John von Neumann is the father of the cellular automata, as I tried to show you. And then it caught my interest. And through Ladenburg, I discovered that his grandfather is Nathanael Pringsheim, biologist, much closer to my heart than quantum physics. I start to get insight in quantum physics, but slowly. Marine biology is closer. That was Pringsheim. Yeah, Pringsheim has the biology, the pattern formation, yeah, artistic approach, culture. So I'm interested by this man personality, the Jewish life. It's very sad to see in this particular case what happened. Yeah, so many aspects. Regarding the model density sensitivity, one way of measuring sensitivity is adhesivity, adhesion. So if the adhesion is strong, you can say it's a higher sensitivity. And adhesivity can be measured in many cellular systems.
More questions? Then I have one. And the last part of your talk was about cancer research. Now, that is becoming a more and more important fight, because since we have start to eliminate other diseases, most of us will go with cancer. And that means that the hospitals will have to deal with lots of cancer cases. So how can your models help the fight against cancer?
Yeah, thanks again. So 12 years ago, my uncle, the nephew of this quantum physicist, he died of glioblastoma multiforme, you know? It's the most malignant brain tumor. And it was exactly my target cancer in which I did research. And I saw that there is a big distance between a patient and this work. So I was very frustrated. However, this has changed, since now I am much more also involved to medical treatments, and I see there is for me not any more difference between a mathematical model approach or an imaging approach. Yeah? You need to do research in all of them. And in cancer, many of the cellular processes are not known. So my work is focused on the cellular processes which contribute to the patient cancer. So this is basic research, yeah? And nobody can say how long it takes from the basic research to applications. Yeah? That's the short answer. And I have over the years been involved in many cancer projects, and I also see treatments which are questionable and so on. So it's, I think, a multi-fold problem. Mathematical modeling is just one methodology which needs to be developed.
Other questions?
I just a question about the patterns in nature. You showed that there are patterns at different hierarchical levels, from genes to individuals. For example, the aggregation of fishes in the fish schools. What about populations? I mean, there are rules, for example, in the metapopulation structure, that is, population get extinct or can grow again according to patterns of gene flow, distance between populations, lifetime of species. There is any rules also on the organization of metapopulations in nature?
So the question is, I was focusing on interactions between agents, and the question was about interactions between populations, so-called metapopulation. There is a very well-developed mathematical theory of metapopulations. Mathematical ecology is one of the oldest mathematician example in biology. So you can see in the history of mathematical biology, it started from actually ecology, and later involved cells, molecules, and so on. And one this is the other side. Yeah, this was a little sub model. One could definitely also include interactions at the population level, but the key point of my presentation was always simplicity. In my cancer model, typically is the question is, why didn't you include nutrients or signal X or Y? Yeah, this can all be done. But the approach here is first to understand the behavior of simple models and then add step by step based on certain questions. Tumor models today, it's a big business. And if you look at them, I would say many of them I call supermarket models. Cancer is one of the probably most complicated, not only complex, but also complicated processes. And you can set up models which have hundreds of variables, parameters. But the question is, it's still a cancer model, sure. But what shall I do with this model? So one has to come from several ends. Thank you.
Are there any questions from home or from Zoom? And speak up. No. Then I thank you very much for your presence here, and I thank the speaker for his discourse. And yeah, then there are here these little goodies. So first, who speaks German? Okay, do you want a German? Anyway, so that's then a given. And then we have the calendars, which have numbers, so you don't need to speak German to understand them. Okay, so thank you very much for your presence here.